Hi everyone, good afternoon. So we're going to be talking about these notes that I took in class over the recombination of bacteria and its application. Alright, so when we're talking about introducing DNA to bacteria, one of the main ways is to transform the bacteria. So we are going to go back to this experiment by Frederick Griffith in 1928, where he injected the mice with strep pneumonia, and it was avirulent. It did not have a capsule in it. Then he's going to add this heat-killed, capsulated virulent strain to the mice. So we're going to also have the heat-killed, capsulated version of this bacteria. And when it was injected into the mice, it showed that the non-capsulated strain acquired the ability to produce a capsule and become virulent. And so later on, Avery, McLeod, and McCarthy would use these other experiments to find that DNA was responsible for that transformation. How could we use transformation? Well, it naturally occurs in the Bacillus and the Neisseria genus. So E. coli could be artificially transformed by this method. You're going to have to resuspend in buffer containing a high level of calcium to bind the DNA to the cell. Then you're going to hold it at 0 degrees, because this 0 degrees Celsius is going to cause the membrane to be brittle. And then when you increase the temperature to 42 degrees Celsius, this causes the DNA to be taken up by the cells of E. coli. This is called heat shock treatment. We could also use electroporation, where we're going to take some of these electrical impulses at high voltage, and they're going to create these pores in the membrane of the bacteria to allow DNA to be taken in. We could also have conjugation. So conjugation was studied by Lederberg and Tatum in 1946, these two scientists. They're going to find the unidirectional transfer from a cell containing a sex plasmid, also known as the F plasmid. This F plasmid is going to be transferred to a cell that does not have the plasmid, aka an F- cell. There's also going to be the transfer of chromosomal genes by conjugation, but this only occurs from the donor cells. The sex plasmid has to be integrated into the chromosome first. The transfer of just the F plasmid to a donor to a recipient occurs with nearly 100% efficiency. So this sex plasmid by itself, the F plasmid, has almost perfect success when it's going from the donor to the recipient. In order to transfer the chromosomal genes, the sex plasmid has to be integrated into that chromosome. We also have transduction, where the infection with a bacteriophage, which is a virus, is close to 100% efficient because the bacteriophage absorbs to a cell surface structure. There's a specific binding there. There is the phage DNA, which is then injected, and then the phage DNA and the capsid proteins are synthesized separately. Remember, they hijack the cell and then they synthesize new viruses. In most phages, the DNA is synthesized as this, a concatemer. A concatemer is a long continuous DNA molecule, and it's going to contain multiple copies of the same sequence linked in series. They're going to be cleaved between the PAC site by ribozymes, so we're going to have some endonucleases, aka some ribozymes. And when this is going to be packaged, it's going to go in, of course, the individual virions. Then the cell will lyse after this whole process. We could use something called shotgun cloning to start with genomic DNA. Then you're going to have a vector that contains a lot of space for DNA, as well as a selectable marker. Usually they use antibiotic resistance. genes. Then you're going to cut the genomic DNA and vector with an endonuclease. Then you're going to ligate it onto these plasmids that you're going to be using as a template. Then you're going to have to transform the E. coli with the plasmid. Then you could also screen for the recombinants via their antibiotic resistance, and this will indicate they have the plasmid. You could also use a restrictase, which will make these sticky ends. This is how they're going to make these like plasmids. Basically, it's like Frankenstein here. We're going to take the restrictase, cut up the genomic DNA, and then you're going to put these pieces, again, it's a random ligation into these plasmids. And then after you've screened with the antibiotic resistance plate, that will indicate if they have plasmid. But then you're going to want to screen for the recombinants for the desired X gene. And this is going to be subcloned out of the gene, out of clone containing X. So you're going to get that desired gene out. Going to re-isolate that plasmid with that specific gene, in lack of better terms. Alright, so we're going to also talk about processing DNA in eukaryotes. We're going to, of course, have to remove the introns. The exons are going to be the express portions of these genetic sequences. So first, mRNA is capped and methyl guanosine monophosphate at the 5' end. That's going to be our 5' methyl guanosine monophosphate cap. And then the poly A tails on the 3' end. You could also splice out the introns. This means that eukaryotic DNA cannot be processed by bacteria, so you would have to remove the introns before transforming any of these prokaryotes. You could use reverse transcriptionase. This enzyme will take the RNA that's been manufactured by eukaryotic processing, and it's going to revert it back to DNA. What's the cool part of this? It's only going to be the exons because we already removed the introns in the RNA template that we're using to make DNA. You could also use degradation with alkali as another method. You could also use DNA polymerase, which is used to synthesize a complementary DNA strand to the single strand. So this is if you're using like a single strand. You could also have the S1 nuclease, and this is going to cut the loop out of the strand. Then once that's finished, you have some double-stranded cDNA, or complementary DNA. Okay, cloning vectors. So we're going to talk about the plasmid. All plasmids have an origin of replication known as an ORI, and then we could also have an antibiotic resistance gene for selection. You usually have several of these antibiotic resistant gene areas, and the restriction sites are also present. The restriction sites that can be cut with different restriction enzymes, so we know what we're putting into these plasmids when we're engineering them. Okay, so first, the plasmid will be isolated from E. coli. Then you're going to cut the restriction enzyme. You're going to use that to cut, create some sticky ends. Then you're going to treat with phosphate. You don't want reforming of this plasmid that you've cut. Then you're going to add the foreign DNA with a restriction enzyme, so BAMH is a restriction enzyme. You're going to also ligate here. Going to ligate into the plasmid these foreign DNA sequences. And then, when they don't grow on this plate with this antibiotic TET, those are the transformed ones. The TET gene was removed, so in this example, the foreign DNA is integrated into the tetracycline resistance gene and therefore disabling it. So, if we have a plate that has this tetracycline on it, and let's say Oh, sorry about that. Let's say we got 5 cells, let's say 5 colonies originally, but one of them doesn't grow when it's placed on the TET plate. So that one right here, this missing one, would have our plasmid. Because when we cut into it, it disabled that region of the resistance gene in the plasmid. We actually also have this ampicillin gene too, so we have another one. Ampicillin, all of them will have this here because we didn't mess with that ampicillin section on the plasmid. So when we went to the TET plate, the tetracycline plate, we know we took out the tetracycline gene from the plasmid and we put something else. Hopefully we put something else there. That's why we have those restriction enzymes, and it offers somewhat of a degree of specificity there. But when we go and plate them on tetracycline, look, we have one of those colonies not there, so that one must have had its gene removed, that plasmid specifically. That's the one with gene X, the gene of interest, this missing colony right here. So we go back to our ampicillin, and then we just get that plasmid out of it. Alright, so now let's talk about preventing religation of an opened vector or a plasmid, an open plasmid. So we're going to be cutting with the endonuclease, right? And then we're going to be treating the ends with a phosphatase. So what does this phosphatase do? This removes the phosphates so it can't reseal. So no phosphates, can't reseal. Foreign DNA is cut with the same endonuclease, and this will provide phosphates used in ligation. So we're kind of recycling these phosphates because of this endonuclease cutting the foreign DNA. Okay, so we could have single and multiple cleavage sites. A single cleavage site in this vector has 3 outcomes. Either the foreign DNA will form a circle here, or the vector reforms, or the vector will incorporate the foreign DNA. However, multiple cleavage sites on a plasmid, this will only result in a small fraction having desired composition, aka only a small amount of these plasmids that we make will have that desired gene X sequence if we try to transform them. The plasmids are convenient but do not work well with large DNA inserts. Alright, so let's talk about some other vectors that do work with larger DNA inserts. So we have the lambda phage. This is a phage with linear DNA about 50 kb long. It's a temperate bacteriophage as well. There are 2 segments that can be edited without phage multiplication being affected. So that means we're not messing with the phage's ability to replicate itself inside a host. These 2 segments are editable, basically. There is an 8 kilobase region of unknown function. This is known as the B region. And an adjacent 7 kb long function that contains at, int, and zis, and these are not needed for the lytic cycle. These 3 regions, that 7 kb. We also have EMBL3 or EMBL3. This is a lambda-derived vector. This insert can be longer than 20 kb or more than the deleted lambda fragments of 15 kb. So in total, we're going to be deleting these 2. However, we have this EMBL3 that we could insert 20 in that space of 15 kb. So the head of the lambda phage can be packaged with a piece of DNA that's slightly longer, about 2 kb, than the length of the normal lambda DNA. This is very useful. More about the lambda phage. The lambda phage particles are produced during the previous stage of infection. The DNA is packaged into these capsids in the cytoplasm of the in the infected cell, the foreign DNA fragment is inserted into the vector DNA by cutting with the restriction endonuclease. Then you're going to anneal, and then you're going to start ligating. When the recombinant DNA is mixed with the proteins that form the phage capsids, the capsid is assembled, and it's going to be packaged into the particles. After packaging, the new phages are going to be used to infect host bacteria with about nearly 100% efficiency. Alright, so now we could talk about the cosmid vector from the amdiphage. The cosmid vector C2RB contains 2 cos sites, and you could edit 40 kilobases, basically. It's a plasmid origin replication is also present there, and we also have a polycloning site. This is a stretch of DNA containing cleavage sites for several restriction enzymes. We also have two antibiotic resistance markers, ampicillin and KAN. Okay, so we're going to cut the cosmid. I should write cosmid up here. We're going to cut with small and BAM-HI to produce 2 cosmic halves. And we're going to insert the 40 kilobase fragments of foreign DNA. This is then packaged and introduced into the E. coli in vitro. They can't be used for proteins that are toxic to the E. coli host, so we can't use them for any products that might be toxic to E. coli. However, they are better than the Hamda vectors for cloning genomic DNA of higher eukaryotes. This is because they can incorporate larger pieces of foreign DNA. Remember, this cosmid, it's 40 kilobases. We also have bacteria artificial chromosomes, also known as BACs. These are plasmid vectors. They have the F factor origin of replication. See that there? They also ensure the plasmid is partitioned into both of the daughter cells. They have these genes here, partition genes, they're called. They're maintained at a very low copy number. So there's like 1 or 2 per cell. So 1, 2 per cell. Just like the F factor. Part of the F factor coding for the cell-to-cell transfer through conjugation has been removed, so it won't spread to other cells. So we kind of had to remove some of this F factor so it won't spread crazily. There's easy isolation from the bacterial chromosome with the artificial chromosome. And we could also, again, it's rare for these to contain more than 1 piece of cloned DNA. We also have derivatives of single-stranded phages. So M13 will infect E. coli cells. However, the product phage within the growing cells, they're going to be producing phage within growing cells without lysis. So no lysis, again, with these single-strand phages. They're going to make this circular single-stranded DNA, and this could be used in a technique called phage display of mutated proteins. The DNA sequence of a foreign protein is inserted into the protein PII, located at the tip of the phage. So we're going to add those mutated proteins and separate them. When the foreign DNA is mutated, each end of the phage particle will display one type of the mutated version on the surface. And we're going to then use an antibody to fish out the mutated phage, because again, they're going to be sticking out. We could use this antibody to check which mutated version of the protein on the surface we want. This mutated phage particle, this protein from the phage particle, when it's mutated, it'll have a different end sticking out, like that, and then we could have an antibody for the one that we want. All right, let's talk about CRISPR. So CRISPR-mediated bacterial immunity. So CRISPRs are genome sequences that defend against invading viruses. They're phage fragments. They're kept by the bacteria. They're composed of the short DNA repeats and spacers. When a virus infects the bacteria, the spacer derived from the virus is incorporated into CRISPR. And you're going to have an unarray with spacers again. The CRISPR RNA sequence is used to transcribe short RNA molecules that associate with the endonuclease Cas. And this endonuclease now has like a targeting system. It's like a missile. It guides them to a target sequence on the evading virus. Then the endonuclease will cut up the DNA. So this is kind of like a rogues gallery. I think we talked about it last time when I took a microbiome class and I was going over notes like this for a review. I called it a rogues gallery basically because it has like this targeting system now. We could use CRISPR for gene editing and gene silencing. So we're going to first design a guide RNA to match the DNA region of interest, the target DNA. Then you're going to have the endonuclease to cut both strands of targeted DNA. Okay, now gene silencing. The cell will attempt to repair the broken DNA, but it results in errors that silence the gene. Okay, so now we go into gene editing. We're going to add a repair template for gene editing that has the change sequence, and it will be incorporated into the DNA of the cell during the repair process. The target DNA is altered so the cell has a new sequence. Again, I wrote it here. You're going to add a repair template with a change in it, and the new sequence will appear in the cells during the repair process. And one of these ways it's being used is a sickle cell anemia, and you're going to be using stems and bone marrow for that process. We could also use clone identification via sequence and probes. So colonies will be grown on agar first, and then you're going to be creating a replica of the colonies on a nitrocellulose filter. The cells are lysed and the DNA is baked onto the filter. So we have this agar with all these colonies. Then we're going to have a replica on this nitrocellulose. So I'm just going to draw X's to show it's like a replica of it. Then you're going to have lysis of the cell, so that's why there are X's there. And the DNA is going to be baked onto the filter. Then you're going to add a probe with fluorescence of the target gene. You're going to have labeled DNA probes, and then you're going to detect binding with autoradiography or fluorescence, not audio, radiography and fluorescence. So it'll light up, and the probe finds a match, and only DNA from this colony hybridized with the probe. All right, that's going to be the last slide, and thanks so much for tuning in. Please do something nice for someone.